Hello, and welcome to today's session on the scientific method. Please be sure to print out the PowerPoint lecture notes before we start. This way you'll have the opportunity to take notes down as we're going through slide by slide. Importantly, you should bring those notes to class. They'll help contribute to the in-class conversation that we're going to have. So let's jump right in and talk a little bit about what science is. And to do that, we're going to start out with a definition that comes to us from the National Academy of Sciences. Okay, so according to the National Academy of Sciences, here's what science is. Science is the use of evidence to construct testable explanations and predictions of natural phenomena, as well as the knowledge generated through this process. So there are two important ideas going on in that relatively straightforward definition. On the one hand, we have something that's very verb-like, and on the other hand, we have something that's very noun-like. We start out with something that's verb-like, that is, an action. Science is the use of evidence to construct testable explanations and predictions. So we're doing something. We're using evidence to construct. There's a lot of action going on there. Okay? Please note that the word testable is underlined. That'll be critical for what we'll be talking about in slides to come. There's also the noun-like portion of this statement, and that is the knowledge generated through this process. Science can be thought of as not just a process, but also as a body of knowledge. And when we think of the body of knowledge interpretation of science, or aspect of science, then we're back to that noun-like aspect. So here it is from the National Academy of Sciences. What we'll do now is try to go beyond just a definition. Definitions are important because they lay the groundwork for our thinking in many cases, but we also have ways of knowing that go beyond definitions. For example, we might go to some criteria that tend to guide science. So here we have a picture of Charles Darwin, very famous for his theory of evolution, and evolution by natural selection. And what we're going to talk about now are three different criteria that tend to guide science. And we'll just go over them first by enumerating them, and then we'll say a little bit more about each of these. So first, we have falsifiability, then we have replicability, and then we have parsimony. And we'll talk about each of these in turn. First, the idea of falsifiability. Is the claim testable? Is it falsifiable? Even in our definition, we saw the emphasis by underlining the word testable, that falsifiability and testability are important ideas for any kind of science, whether we're talking about the science of psychology or the science of biology, chemistry, physics, geosciences, astronomy, whatever the case might be. Testability is going to be absolutely key. Sometimes students are thrown by this word falsifiability, so I'd like to say just a little bit more about that. Let me turn down this image and we'll draw a diagram that will help us understand falsifiability a little bit further. Okay, so what we can say is that we want to create something like a Venn diagram that will help us to understand what falsifiability is. And we're going to draw a very large circle. Okay? And what we're going to say is in the center of the circle, we're going to have the word false. And we'll circle that. And then this larger circle will be the circle of falsifiability. Or that which is falsifiable. Okay? So there are some claims that people might make that are outside of the sphere of that which is falsifiable. These might be, for example, faith-based claims. And lots of folks will find that to be an important aspect of their life, their faith, but that's beyond the domain of science. When we're making claims that scientists would be interested in, we're interested in specifically falsifiable claims that will land somewhere in here. A subset of those falsifiable claims might be shown by observation or by experiment to be false. Okay? And if you're telling me that something is false, you are implicitly telling me that something is falsifiable. Okay? So to say that something is false is not necessarily the same thing as, or excuse me, is in fact to imply that something is falsifiable. But if you're telling me that something is falsifiable, you haven't gone to the next step and shown me that in fact it is indeed false. One other example that might help you. Many of us know how countries land within particular kinds of continents. So if you tell me that somebody is living in the United States, you are implicitly telling me that they're somewhere in North America. On the other hand, if you tell me that somebody is living in North America, it may or may not be true that they're living in the United States. That same kind of relationship exists between that which is false and that which is falsifiable. And sometimes students need a little bit of practice to getting used to that particular distinction that we're making here. Okay, let's take this away, and we'll go back to our presentation. Okay, and we'll get to the idea of replicability. Can a test be done and redone at will? This is the notion of replicability, and in the slides to come, we'll be talking at some considerable length about the issue of replicability with respect to Darwin's theory of evolution, just to give us a specific example to understand this notion of replicability and why it's important to scientists. Lastly, we have parsimony. Are there unnecessary or unnatural components in an explanation? We try to get rid of those. Some of you may have taken a course in philosophy, and if you did, you might have heard about a particular philosophical idea called Occam's razor. Occam's razor is can be boiled down to a fairly simple statement which goes something like this. All other things being equal, the simplest explanation is the best. I'll say that again. All other things being equal, the simplest explanation is the best. That's Occam's razor, and that embodies this idea of parsimony. Are there unnecessary or unnatural components? Science does not get involved in claims that are supernatural or beyond the bounds of space and time. Some faith will take us beyond space and time, but science operates very much within the domain of space and time, so we won't have any unnatural components, and we're looking for ideas that are expressed parsimoniously. That is, they don't contain anything that they don't need to contain. You might think of this in a way as being a type of explanatory economy. We want very economical kinds of expressions. In this sense, we're not using economics to refer to money. We're using the word economical to refer to the idea that we don't have anything more than what we need. Now, some people might say, why do we bother with that? Why do we bother with parsimonious explanations in psychology and in other sciences as well? And really the answer is twofold. On the one hand, we can say that that's simply a stylistic difference that distinguishes the sciences from other ways of knowing, maybe artistic ways of knowing that value aesthetics very strongly. Maybe there are religious ways of knowing. Our style is one that is parsimonious. And you might ask, well, why do you have a parsimonious style? And interestingly, we can answer that question by referring to simple probability. So once again, I'm going to turn down this image and give us a few diagrams here that I'll draw on the board that will help us to understand why it is the case that parsimony is very much related to probability. Okay. So here we are talking again about parsimony, and the issue becomes how is this related to probability? So let's take a specific example that might be most relevant to psychology. Let's say that you are interested within the domain of psychology in the phenomena of depression. Depression is a naturally occurring phenomena. It's actually the source of much human suffering, so it's an important problem. And let's say that you're trying to explain the phenomena of depression, and you have a relatively parsimonious explanation for depression. For simplicity, we'll say that your theory or your hypothesis about depression is that depression arises because of neurotransmitter X, whatever X might be. Okay? So you're saying that neurotransmitter X is the cause of depression. So this is, if you will, hypothesis A is saying that it's neurotransmitter X. That's at the root of depression according to one theory. Let's say I have a theory also, and my theory is similar to your theory, but it's less parsimonious. Let's say that I claim that depression arises not from neurotransmitter X alone, but from neurotransmitters X and Y. So now I have my own theory, theory B, and I've got the same kind of idea, still neurotransmitter, but now it's X and Y. I'm positing two explanatory components. In this case, it's two neurotransmitters. You're positing just one. Okay? All other things being equal, we'll pretend that your explanation explains as much about depression as mine does. Yours is more parsimonious. We would favor yours over mine because mine has an additional explanatory component. Now, why might that be the case related to probability? We'll use the simple example of a coin flip, since most of us have some experience with that. And we know that when we flip a coin, we colloquially say that we get either heads or tails. And we'll say that the probability of a head is 0.5, the probability of a tail is 0.5. So we're now asked the question, what is the probability that neurotransmitter X really is the explanation for depression? And just for fun, we're going to pretend that you've got a 0.5 probability. Okay? So maybe you're right, maybe you're wrong. The probability in this hypothetical case, very much simplified, is 0.5. Okay? Now let's take a look at my hypothesis. I've got one that's stipulating neurotransmitter X and Y. We've already established that the probability of this component being true has a probability of 0.5. Okay? And for simplicity, we'll also say that my neurotransmitter Y component has a probability of 0.5 as well. In probability theory, what we typically do is we want to say that if we want to know the joint probability of two events, X and Y, we have to multiply these one by the next. And as you might remember from your earlier days working with decimals, 0.5 times 0.5 equals 0.25. Okay? So now the joint probability of neurotransmitter X and neurotransmitter Y being relevant to depression is going to be 0.25 versus your probability of 0.5 alone. So all other things being equal, yours is a more probable explanation than mine is, because you have a much higher you have a probability of 0.5 and I have a probability of 0.25. So this, I hope, gives you some intuition about how the idea of parsimony is related to the idea of probability. Okay, let's take this away and we'll bring back our next slide. Here we'll talk about what science is with respect to different virtues that characterize science. And I think an important point about this is to recognize that the virtues that we're going to call out in just a moment might exist in gradations. It's not that we have an all or none scenario here. We have varying degrees to which we recognize these virtues in any given theory or any given experiment. The same thing was true, by the way, in the previous slide when we talked about falsifiability, and we talked about replicability and parsimony. Those exist in gradations, and that's an important aspect of how we think about the different scientific criteria and the different scientific virtues. Let's go here and first give you a very simple memory device. Many people who are in the United States are familiar with a common acronym, GOP, which stands for the Grand Old Party in the political sphere. During today's conversations, we're not talking about politics, we're talking about science, and we're going to use GOP in a different way. We're going to talk about the generalizability, the originality, and precision of a given scientific endeavor. And to motivate our conversation a little bit further, we'll stay with Darwin, and we'll stay with Darwinian evolution, and we'll talk about the generalizability, the originality, and the precision of Darwin's theory of evolution by natural selection. Okay, let's take generalizability first. When we're talking about generalizability, we're talking about the extent to which the findings or claims that are made can be realized in novel settings and novel environments and so forth. So what we're going to do is think about generalizability with respect to Darwin's theory of evolution and remind ourselves that when Darwin was a young man, probably not too different in age from most college students, he went to the Galapagos Islands in December of 1831, and he began making many, many observations about plant life and about animal life. And he was trying to explain the diversity of life, and he came up with a theory that you're probably very familiar with. If we're talking about the generalizability of that theory, we can ask this question. To what extent did Darwin's theory generalize beyond the Galapagos Islands where he first made those observations? Had it been true that his theory was relevant only to the Galapagos, then we might say that his theory had relatively limited generalizability. On the other hand, if his theory pertained to other islands as well, then he has more generalizability. if his theory pertains to structures that are not just islands, but to deep sea life and to larger land masses. In fact, if his theory is true everywhere in the world, then he has terrific generalizability. And in fact, that's what we actually experience with Darwin's theory of evolution. It generalizes, as far as we can tell, to life anywhere that we can find on the planet, whether it's in the cold extremes that we have on the polar caps, or in deep sea life, or on land. No matter where we go, we find that his theory of descent with modification, evolution by natural selection, generalizes very well. So he gets high marks on generalizability. Let's go to originality. An important synonym here might be novelty. To what extent was his idea a novel one and an original one? And people might quibble a little bit about this. There's a fairly well-known researcher from his day, actually a contemporary of Darwin, also from the United Kingdom. He was from Wales. His name was Alfred Russel Wallace, and he had similar ideas to Darwin. So it wasn't that Darwin was the only one who had these ideas, and people before these two gentlemen would have had perhaps similar ideas, but Darwin was really the one who maybe had amassed the most evidence and told the most coherent story in his document on natural selection in 1859. It was really quite original, and to give you some idea about how original it was, there had been terrific biologists prior to the time of Darwin, including all the way back to Aristotle, who had a lot to say about the diversity of life. But he wasn't able to come up with a theory of evolution, which very elegantly explains the diversity of life. We can think of other great scientists, including Sir Isaac Newton, who was so clever that he had actually discovered and began different branches of physics. And when he needed more mathematics to describe the branches of physics that he himself was discovering, he just developed the new branches of mathematics. He developed certain forms of calculus that were not known prior to his thinking. Clearly a very brilliant scientist, and yet was unable to come up with something like the relatively elegant solution that evolution is in the world of biology. So clearly a very original idea. One further notion about the originality of Darwin's theory of evolution is that even today, many people will hear about the theory of evolution and they won't believe it. And that is because it's perhaps difficult to imagine how it is that you and I as humans have a common ancestor with something like an oak tree or a bacterium. It's not very obvious, it's not very intuitive. It would take a lot of originality to create something like that, so Darwin's theory of evolution gets high marks for originality. Let's now move over to precision and talk about how that might relate to Darwin's theory. When we're describing precision, we're talking about the fineness of a measure or the fineness of a prediction. To what extent can a prediction be laid out maybe in time or laid out in space in very precise or very small intervals of time or small intervals of space? And what's nice about Darwin's theory is it makes very, very precise predictions on lots of different dimensions. Let's talk about some of these. First, Darwin's theory would make predictions about the kinds of fossils that we might find as we're looking about the Earth. And what we've learned since his day is that we do find all kinds of fossil evidence that's very consistent with the timeline that he had laid out. We see lots of transitional forms from this type of an animal to that type of an animal. Very persuasive because it's very consistent with his timeline. Another idea about the precision of his theory is that the theory makes very precise predictions with respect to the geographic distribution of plants and animals. You might notice that there's a very non-random distribution of plants and animals, and all of that non-randomness is highly consistent with his theory of evolution. Finally, and I think maybe most spectacularly, we can look at what we now call today molecular evidence, molecular genetics. We can look at something that was not even known in Darwin's time. Darwin wrote this piece about the theory of evolution in 1859, or that's when he published it anyway. It wasn't until 1953 that Crick and Watson had found the molecule that we call DNA. So about 100 years later, 90-something years later, we find this molecule called DNA. And now we can begin to use this new technology to test the precision of Darwin's predictions. For example, Darwin would have predicted that we would be more similar to chimpanzees than we are to, say, some other mammal, like maybe a dog or a cat. And if we now look at the molecular evidence, the patterns of G's and C's, and A's and T's, these nucleotides, we do see much greater genetic similarity between humans and chimps than we do between humans and, say, dogs or cats. Okay? So there's a great precision in the sequence that he had laid out, and this is why he gets high marks for precision also. Okay? All right, so this is our notion about why it is that we have these virtues. We have, again, the GOP, the generalizability, the originality, and precision of any given scientific theory or maybe a particular work of science. Why don't we take a pause right here? I'll ask you to stop the video and make some notes to yourself about what you find particularly interesting or compelling about this, or maybe some questions that you have or doubts that you have about this. And I look forward to hearing those in class.